She was 13 years old when she died, and for 90,000 years after that, she was nothing more than a splinter of bone in a plastic tray, sitting in a drawer with 2,000 other splinters that nobody could identify. No skull, no face, no grave marker. Just a piece of arm or leg, 2.5 centimeters long, pale and unremarkable, the kind of fragment a researcher might glance at for 3 seconds before setting it aside. For 4 years it sat in that drawer in a laboratory in Siberia, catalogued only by a number. Denisova, 11. Nobody knew it belonged to a girl. Nobody knew it belonged to someone who should not, by every rule scientists understood about the ancient world, have existed at all. Her mother was a Neanderthal. Her father was something else entirely, a different kind of human being, so rare that scientists had only ever found a handful of his kind, always in the same cave, always in fragments so small that an entire species had been built from a finger bone and a tooth. 2 ancient human populations, separated for hundreds of thousands of years, evolving apart on different sides of a continent, developing different bodies, different faces, different lives, had somehow found each other in the mountains of southern Siberia, and they had made a child. That should not have been possible in the story most people had been taught about ancient humans. For most of the last century, extinct human relatives were treated as separate branches on a family tree, dead ends, failed experiments that vanished when modern humans arrived, and did what modern humans apparently did best, which was survive. Neanderthals were a footnote, a different species entirely, distant cousins who went extinct while we did not. The idea that a Neanderthal woman could stand in a cave beside a man from an entirely different, barely known population, and that these 2 people could fall into whatever arrangement produces a child, was not part of the story anyone expected to prove. Then, in a laboratory in Germany, a bone fragment the size of a fingernail gave up a secret that rewrote the ending. This is the story of a girl who lived and died in a cave carved into a Siberian cliff, roughly 90,000 years before anyone would ever write her name, because nobody knew her name and never will. It is the story of 2 parents from 2 different kinds of humanity, who met somewhere in the shadow of the Altai Mountains and produced a daughter whose bones would eventually prove that the line between species was never as solid as it looked. It is a story about a cave that has been called one of the most important places on Earth for understanding what it means to be human, and about the scientists who spent years trying to squeeze meaning out of fragments too small to hold in a closed fist. Her discoverers eventually gave her a nickname. They called her Denny. Before we go further, a question worth sitting with for a moment. In your own family, has anyone ever traced their ancestry and found a surprising mix of backgrounds coming together in one person? Something unexpected, buried a generation or 2 back, that only came to light through a DNA test or an old family story. Keep that thought close, because Denny's discovery began the same way every family discovery begins. Somebody looked closely at something ordinary and found a story nobody expected. Denny was found in Denisova Cave, a limestone cavern set into a river valley in the Altai Mountains, a place cold enough in winter to freeze exposed skin within minutes, and green and alive in the short mountain summer. The cave takes its name today from her father's people, the Denisovans, but for most of its history, it belonged to no single group. Different populations of ancient humans moved through it, occupied it, left behind their tools, their fires, their broken bones, sometimes for a season, sometimes for centuries, and then moved on or vanished, leaving the next occupants to walk over ground that already held the remains of a people they never knew. By the time archaeologists began serious excavation there in the 20th century, the cave floor held more than 3.5 meters of accumulated sediment, layer stacked on layer, each one a slice of time going back further than the last. Somewhere near the bottom of that stack, in a layer that has never been precisely dated but is understood to be extremely old, someone left behind a small piece of bone. It came from an arm, or possibly a leg. It was so badly broken and so visually unremarkable that when archaeologists pulled it from the ground in 2012, nobody looked at it twice. It went into a tray with roughly 2,000 other bone fragments too small and too damaged to identify by eye. Most of those fragments were animal. A handful, buried among the rest, were not. The fragment sat unexamined for years, because there was no realistic way to know which of 2,000 splinters belonged to a person and which belonged to a reindeer or a bison. The breakthrough that eventually separated them came not from a paleontologist squinting through a microscope, but from a technique borrowed from crime laboratories and quality control departments. Collagen, the structural protein that gives bone its strength, differs slightly between species in ways that a method called peptide mass fingerprinting can detect from almost nothing. A research team working through the tray of unidentified fragments used this method to sort bone from bone, animal from human, without needing to recognize a single visible feature. Somewhere in that sorting process, a research group led by a scientist named Samantha Brown flagged one small fragment as belonging to an archaic human. That flag was the first sign that Denisova 11 was more than driftwood in a drawer. Once it was identified as human, it went to a laboratory built for exactly this kind of work, a facility in Leipzig, Germany, run by a research team that had spent years perfecting the extraction of genetic material from bones so old and so degraded that most laboratories in the world would consider them a lost cause. The team was led by a scientist named Svante Parbo, a name that by then had already become inseparable from the modern understanding of ancient human DNA, and a geneticist named Vivian Slon, who took the lead on sequencing this particular fragment. What came out of that sequencing did not simply identify Denisova 11 as human. It identified her, almost immediately, as something that had never been documented before. Her genome carried two distinct genetic signatures in roughly equal measure, one matching Neanderthal populations, the other matching the Denisovans, the mysterious population first identified from that same cave only a few years earlier. A geneticist reading a genome expects to see the ancestry of a population, blended slowly over many generations, shaped by centuries of intermarriage and migration. What Slon's team was looking at was not that. It was a 50-50 split, so exact that there was only one explanation. This was not a descendant of a mixed population. This was a child, a first-generation child, born to one parent who was fully Neanderthal and one parent who was fully Denisovan. Nothing like it had ever been found. Not once, in all the archaic human remains ever excavated anywhere on Earth. Scientists had proven, years earlier, that Neanderthals and Denisovans had each contributed genetic material to the ancestors of people alive today, evidence that both groups had interbred with modern humans at some point in the deep past. But that evidence came from small percentages scattered through modern genomes, the genetic echo of unions that happened thousands of generations back, diluted and reshuffled by time. What Slon's team had in front of them, in the case of Denisova 11, was not an echo, it was the moment itself, a direct genetic snapshot of a Neanderthal mother and a Denisovan father, standing close enough, together long enough, for a daughter to result. The paper describing the discovery appeared in August 2018 in the journal Nature, and it did something rare for a scientific publication. It made the front pages of newspapers around the world within a day. An anthropologist at the University of Wisconsin-Madison, John Hawkes, who had no part in the study, was quoted at the time describing the discovery as a story more remarkable than anyone studying ancient interbreeding had expected to actually find. He was right. A single bone fragment, plucked almost by accident from a tray of debris, had done what years of theorizing about hybrid populations had never managed. It had produced direct, undeniable proof. But a genome is not a person. It tells you what happened in a cell. It does not tell you what happened in a life. To understand who this girl actually was, and why her existence matters so profoundly to the story of human origins, we have to go back further than her birth. We have to understand her mother's people and her father's people, two populations who had been separated for hundreds of thousands of years before anything brought them back together in the same stretch of Siberian mountain. We have to understand the cave that held her secret for 90,000 years, and the strange, quiet drama of two very different kinds of human beings finding themselves, somehow, close enough to become a family. This is that story. Chapter 1. The Woman Who Came from the West. To understand Deni's mother, you have to travel back across a continent, and back across time, to a species that had been quietly perfecting survival in the coldest parts of Eurasia for hundreds of thousands of years before her birth. Neanderthals are, by now, the most familiar of all extinct human relatives, but familiarity has not always meant respect. For most of the 20th century, the word conjured a caricature, a hunched, brutish figure, dragging a club, incapable of speech or thought beyond the next meal. That image came from a mixture of poor early reconstructions, cultural bias, and a genuine, if unfair, human need to see modern people as the pinnacle of a long climb upward, with everyone else along the way cast as an inferior draft. Modern research has spent decades correcting that picture, and what has replaced it is a population far more capable, far more adaptable, and far more human than the caricature ever allowed. Neanderthals lived across a vast stretch of territory, from the Atlantic coast of what is now Portugal and Spain, through Central Europe, into the Near East, and eastward through Central Asia, as far as the mountains where Denisova Cave sits today. They were not a small, isolated population, clinging to survival in one corner of the world. They were spread across an entire hemisphere of shifting climate, from temperate forest to open steppe to the edge of glaciers, and they endured that range of environments for a span of time that dwarfs the entire history of modern civilization many times over. Physically, Neanderthals were built for cold and for strength. Their bodies tended to be shorter and more compact than the average build of modern humans, with wide rib cages, strong limbs, and heavy brow ridges over deep-set eyes. This was not primitive design. It was efficient design, the kind of body that conserves heat, generates power for close-range hunting, and absorbs physical stress without breaking. Their brains, measured by the space inside their skulls, were on average as large as those of modern humans, sometimes larger. Whatever intelligence requires in terms of raw neural tissue, Neanderthals were not short of it. The archaeological record left by Neanderthal populations tells a story of skill and adaptation that took decades to fully appreciate. They made sophisticated stone tools, shaped through a technique that allowed them to control the size and edge of a blade with real precision. They hunted large, dangerous animals at close range, a task that required coordinated group effort, patience, and courage, not blind aggression. Evidence from sites across Europe shows they used fire not only for warmth but for cooking, for hardening the tips of wooden spears, and likely for processing certain plants to make them digestible. Chemical residue on their teeth, preserved for tens of thousands of years, has shown that their diet was not the meat-only stereotype long assumed. Some populations consumed a wide range of plants, seeds, and even what researchers have interpreted as evidence of self-medication using specific plants with anti-inflammatory properties, found in the dental plaque of Neanderthal individuals who appear to have been suffering from illness. Perhaps most significant to the story of who Deni's mother was as a person, rather than simply a biological category, is the growing body of evidence that Neanderthals cared for their sick, their injured, and their elderly. Skeletons have been recovered showing individuals who survived for years after injuries that should have been fatal without sustained help from others. A skeleton found in Iraq from an individual known to researchers as Shanidar 1 shows a man who lived for a significant period after losing the use of one arm and suffering severe damage to one side of his skull, injuries that would have made him unable to hunt or fully provide for himself. He survived anyway, which means someone fed him, protected him, and kept him alive long after his usefulness to the group, in a purely practical sense, had ended. That is not the behavior of a brutish, instinct-driven animal. That is the behavior of a society that valued its members beyond their immediate output, a behavior we recognize immediately in ourselves, because we still argue, in every generation, about what a community owes to those who cannot fully care for themselves. Neanderthals also buried their dead, in at least some cases with what appears to be intentional care, bodies placed in deliberate positions, sometimes accompanied by objects that may have held meaning beyond the practical. Whether this constituted belief in an afterlife, or something closer to grief and ritual without a formal religious framework, remains genuinely debated among researchers, and it would be dishonest to claim certainty either way. What is not debated is that the placement of bodies in many of these cases shows intention. Someone chose to arrange the dead in a particular way. Someone returned to a specific location to do it. That points toward memory, toward relationships that outlasted a single life, toward something recognizably close to what we mean by loss. The population that eventually produced Deni's mother was part of a Neanderthal lineage that had, at some point, moved eastward, deep into Asia, following game, following rivers, following whatever combination of pressure and opportunity moves any population across a landscape over many centuries. Genetic analysis of Deni's own genome and of other Neanderthal remains found in the same cave has allowed researchers to trace something remarkable about her mother's ancestry. Her mother's genetic makeup was more closely related to Neanderthal populations who lived later in Europe than it was to an earlier Neanderthal individual whose remains were also found in Denisova Cave itself. In simple terms, the woman who gave birth to Deni was not descended from the Neanderthals who had lived in that same cave generations earlier. She came from a different, more Western lineage, one that had made its own long journey east. This detail matters more than it might first appear. It means Deni's mother's own ancestors, or perhaps her mother herself, undertook a migration across enormous distances, potentially thousands of kilometers, moving through unfamiliar territory, adapting to new landscapes, encountering populations along the way that her own ancestors had never known. Neanderthal movement across Eurasia was not a single event. It was likely a series of gradual population shifts, families and small groups moving with the seasons, sometimes over many generations covering ground that no single individual would ever fully retrace. The Neanderthals of Denisova Cave, then, were not one static population sitting still in one location for hundreds of thousands of years. They were part of a living, moving, connected world of related groups, spreading and contracting and reconnecting across an entire continent. And Deni's mother was one thread in that enormous, largely invisible tapestry of ancient movement. What we do not know, and cannot know, is anything about her as an individual. We do not know her name, because no name survives from a species that likely never wrote anything down. We do not know her face, her voice, her personality, whether she was cautious or bold, whether she was the kind of person others in her group turned to for guidance, or a younger woman still learning the skills that kept a small band of people alive through a Siberian winter. What we know is genetic, structural, and circumstantial. She was Neanderthal. Her lineage had crossed vast distances to reach the Altai Mountains, and at some point in her life, in or near a cave that still stands today, she came into contact with a population unlike her own. Chapter 2: The Mystery in the Other Direction. If Neanderthals are the well-documented branch of Denis' family tree, her father's people are the opposite. The Denisovans remain, even now, one of the strangest and least understood populations ever identified in the story of human evolution, a group defined almost entirely by absence, known through scraps rather than skeletons. The story of how Denisovans were discovered at all begins with a single small bone, found in the very cave that would later give up Denis' remains. In 2008, archaeologists working in Denisova Cave recovered a fragment of a finger bone from a layer of sediment. It was so small and so unremarkable that, on its own, it told researchers almost nothing. What made it extraordinary was what came next. When the fragment was sent for genetic analysis in 2010, the DNA extracted from it did not match modern humans. It did not match Neanderthals either. It matched something else entirely, a population that had never been identified before, related to both modern humans and Neanderthals, but distinct from each. This was an almost unprecedented moment in the study of human origins. An entire, previously unknown branch of the human family tree had been identified not through a skull, not through a skeleton, but through a bone smaller than a fingernail, using genetic technology that simply had not existed a decade earlier. The population was named after the cave where the finger bone was found. Denisovans. In the years since that discovery, the physical evidence for what Denisovans actually looked like has remained almost shockingly thin. Beyond the original finger bone, researchers have identified only a handful of additional fragments confidently attributed to the species. A few teeth, unusually large and robust compared to modern human teeth. A fragment of skull, a single fossilized jawbone, found not in Siberia but thousands of kilometers away, on the Tibetan Plateau in China, in a cave known as Baishia Karst Cave. That jawbone has its own remarkable story. It was found in 1980 by a local Buddhist monk, who discovered it inside the cave and eventually passed it to religious authorities, who in turn eventually connected it to researchers decades later. For years, nobody could determine what species it belonged to, because the specimen contained no preserved DNA that scientists could extract using the technology available at the time. It took a specialized technique called ancient protein analysis, examining degraded proteins locked inside the tooth enamel rather than DNA, to finally confirm in 2019 that the jawbone belonged to a population closely related to the Denisovans of Siberia. Later, in 2020, researchers succeeded in extracting genetic material directly from the sediment of the cave floor itself. Not from a bone, but from the dirt the population had lived and died on, confirming Denisovan presence at that high-altitude site, across a span running from roughly 100,000 years ago to as recently as 45,000 years ago. This detail, that Denisovans were living not only in the frozen valleys of Siberia, but also on the Tibetan Plateau, at an altitude above 3,000 meters, thin on oxygen, and brutal in climate, tells us something important about who they were as a population. Modern humans who live at extreme altitude today, including Tibetan populations, carry a specific genetic variant that helps their bodies process oxygen more efficiently in thin mountain air. Researchers have traced that exact genetic variant back to Denisovan ancestry. In other words, a population once dismissed as an obscure genetic footnote left behind a survival adaptation so valuable that it is still, today, helping millions of people breathe more easily at high altitude. That single fact turns Denisovans from an abstract curiosity into something with a direct, measurable legacy in living human bodies right now. Because so little physical evidence survives, reconstructing what Denisovans actually looked like has depended heavily on genetic modeling rather than direct observation. In 2019, a research team used patterns of gene activity comparing Denisovan DNA against the DNA of species with known physical traits to predict likely Denisovan characteristics. Their model suggested a population with a wide skull, a low and sloping forehead, a large jaw, larger teeth than modern humans, and a barrel-shaped rib cage, similar in some respects to Neanderthals, though not identical. Researchers have been careful to describe this as an informed prediction rather than a confirmed portrait, because without more complete skeletal remains, absolute certainty about their full appearance is simply not available. What can be said with more confidence is that Denisovans, whatever their precise appearance, likely bore a strong resemblance to Neanderthals in overall build, while carrying their own distinct features shaped by a different evolutionary path. Genetically, Denisovans and Neanderthals share a common origin. Both populations descended from an earlier human lineage that split away from the ancestors of modern humans roughly 600,000 years ago, migrating out of Africa and into Eurasia. At some point after that migration, the ancestors of Neanderthals and the ancestors of Denisovans separated from one another, likely somewhere in Western Asia, with one branch moving toward Europe and the other moving deeper into Eastern Asia. Genetic estimates place that split at somewhere between 390,000 and roughly 500,000 years ago, depending on the specific method used. From that point forward, the two populations evolved largely apart, adapting to different environments across an enormous span of time, developing along separate paths for hundreds of thousands of years. And yet, remarkably, they never lost the ability to produce children together. This is one of the most genuinely astonishing aspects of the entire story. In the modern biological world, different species are usually defined, at least loosely, by their inability to produce fertile offspring with one another. Horses and donkeys can produce a mule, but the mule cannot reproduce. Different bird species may occasionally hybridize, but the results are often sterile or rare. Neanderthals and Denisovans, separated for a span of time longer than the entire existence of modern civilization many times over, apparently retained full biological compatibility. Whatever divided them, whatever different lives they had built across hundreds of thousands of years apart, it was not enough to prevent them from having children when circumstances brought them together. That fact alone forces a reconsideration of how rigidly we should think about the boundaries between these ancient populations. They were different. They were not incompatible. Beyond the physical and genetic evidence, researchers have also worked to understand something of Denisovan behavior and technology, largely by examining tools and animal remains found in layers of Denisova Cave and by sheer karst cave associated with their occupation. The evidence suggests a population capable of sophisticated tool production, similar in general capability to Neanderthals and early modern humans, including implements made from bone as well as stone. Excavations at Sheer Karst Cave have uncovered animal remains, including the bones of blue sheep and other mountain mammals, showing signs of butchery, suggesting a population skilled at hunting and processing large animals in an extremely demanding high-altitude environment. This was not a marginal, barely surviving population. This was a group that had found a way to thrive in one of the harshest inhabitable environments on the planet, generation after generation, for well over 100,000 years. What remains almost entirely unknown is anything resembling the individual identity of any single Denisovan person. We do not know their language, if they had one in a form we would recognize. We do not know their social structures, their beliefs, their family patterns. We know they existed, we know roughly where, we know roughly when, and we know, because of Denny, that at least one Denisovan man and at least one Neanderthal woman found each other close enough and stayed together long enough for a child to result. Somewhere in the vast, mostly silent record of the Denisovan past, there was a father, a man whose genome, when scientists eventually read it inside his daughter's bones, would reveal something else unexpected. Even he was not free of mixture. His own ancestry carried a small trace of Neanderthal blood, inherited from an encounter that had happened centuries before his own birth, an encounter separate and distinct from the one that produced his daughter. His people and hers had, it seems, been finding each other for longer than anyone suspected. Chapter 3. The cave that kept everyone to understand why Denny's bone fragment ended up in Denisova Cave at all, and why that cave has produced more insight into ancient human interbreeding than almost any other site on Earth. We need to understand the place itself, not as a backdrop, but as a character in this story, with its own long, strange history. Denisova Cave sits in the Altai Mountains of southern Siberia, in a river valley carved by the Inui River, at an elevation that places it firmly within a landscape of forested slopes, sharp winters, and short but productive summers. The cave takes its modern name from a Russian hermit named Denis, who is said to have lived there in the 18th century, centuries before anyone understood what lay buried beneath the cave floor. Long before Denny, and long before that, the cave had already been serving as shelter, workshop, and burial ground for a rotating cast of ancient human populations stretching back further than almost any other site of its kind. The cave's true significance only became clear through decades of careful excavation, much of it led by Russian archaeologists working through the site's sediment layer by layer. What they found, over time, was not a simple record of one population living in one place. It was something closer to a layered history book, written not in ink but in stone tools, animal bones, ash from ancient fires, and eventually genetic material preserved in the very dirt of the cave floor. Sediment samples taken from different depths and different chambers have allowed researchers to identify occupation by multiple distinct human populations across an almost unimaginable span of time, with archaeological evidence at the site reaching back roughly 300,000 years, and possibly further. Denisovans were present in the cave for a large portion of that span, based on the layers where their fossils and genetic traces have been recovered, with sediment evidence indicating a Denisovan presence stretching from around 287,000 years ago through to more recent periods. Neanderthals also occupied the cave at various points, their remains and genetic traces appearing in different layers, sometimes overlapping in time with Denisovan occupation, sometimes not. Later still, modern humans arrived in the same valley and left their own traces in the uppermost layers of the site. 3 distinct human populations, each returning to the same shelter across vast stretches of time, sometimes generations apart, sometimes, as Denny's existence proves, close enough in time and space to actually meet. The scale of that time depth is difficult to hold in the mind honestly. If you imagine the entire span of the cave's known human occupation as a single day, modern written history, everything from the earliest civilizations to the present moment would occupy only the last few minutes before midnight. For nearly the entire span of that imagined day, the cave belonged to populations who left no writing, no cities, no monuments beyond broken stone and ash, and yet who lived complete human lives, raised children, buried their dead, and in at least one remarkable case, produced a daughter whose existence would eventually prove that the boundaries between these populations were far more permeable than anyone assumed. Excavation at Denisova Cave has produced an extraordinary volume of material. Researchers have recovered tens of thousands of stone tools and animal bones from the site, evidence of sustained, repeated habitation across an enormous time span, alongside items of surprising sophistication. A bracelet made of polished dark green stone, recovered from a layer associated with early modern human or possibly late archaic human occupation, shows evidence of drilling and polishing techniques that some researchers have argued represent surprisingly advanced craftsmanship for its age. Whether every detail of that specific find's dating and attribution remains fully settled among specialists, the broader picture from the site is consistent. This was not a marginal, forgotten corner of the ancient world. This was a place people kept returning to, generation after generation, population after population, because it worked. It offered shelter from Siberian winters that could otherwise kill an exposed person within hours. It sat near a river that provided water and likely fish. It sat within reach of forest and open steppe, giving access to a wide range of game animals across different seasons. In simple terms, Denisova Cave was valuable real estate in the ancient world, and valuable real estate tends to attract more than one group of people. When multiple populations share access to the same limited number of genuinely good locations across a landscape, across enough time, the odds of overlap increase. Not constant contact, not necessarily even frequent contact, but enough contact, across enough centuries for paths to cross. This is one of the central insights that Denis' discovery, and the broader body of research on Denisova Cave, has forced onto the scientific understanding of ancient human geography. For a long time, the guiding assumption was that Neanderthals and Denisovans occupied largely separate territories, Neanderthals dominant across Europe and Western Asia, Denisovans confined to Eastern Asia, with limited overlap between them. The cave complicates that picture considerably. If both populations were regularly present in the same valley, in the same cave system, across overlapping stretches of time, then the border between their territories was not a hard line on a map. It was a shared, contested, sometimes overlapping frontier, a place where two different kinds of humanity were, at various points across tens of thousands of years, effectively neighbours. Being neighbours does not automatically mean warmth, or partnership, or family. It can mean competition for the same resources, tension over the same shelter, avoidance rather than connection. The archaeological record cannot tell us, with certainty, what the general relationship between these populations looked like across the full span of their overlap in this region. What it can tell us, through Denis' own bones, is that at least once, in at least one case that we happen to have found evidence for, the relationship moved past avoidance and competition into something that produced a child. There is a sobering point worth acknowledging honestly here, one that the researchers who studied Denis' genome themselves raised. We do not know, and cannot know, whether the union between her parents was something we would recognize today as a relationship built on mutual choice, or whether it reflected the harsher realities that likely existed between different, sometimes competing ancient populations. The genetic record proves that her parents came together. It cannot tell us the full circumstances of how, or why, or under what conditions. Responsible science holds that uncertainty rather than filling it with an easy, comforting assumption, and it is worth sitting honestly with the limits of what bone and DNA alone can reveal about the human choices, or lack of choice, that shaped a life 90,000 years ago. What we can say, based on the layer in which Denis' remains were found, and based on genetic dating techniques applied to her own DNA, is roughly when this meeting occurred. Her genome allowed researchers to estimate her age at death, and, working backward through genetic mutation rates to estimate roughly when her parents lived. The evidence places her mother's generation at approximately 90,000 years before the present, meaning Deni herself likely died around that same period, a young teenager whose short life ended in a place that, whatever else it was, had clearly been significant enough to her community or her family to serve as her final resting place. Chapter 4. The Splinter That Waited in a Drawer. The physical journey of Deni's bone fragment from the ground to a laboratory desk in Germany is, on its own, a story about patience, persistence, and the strange, quiet role of chance in scientific discovery. The fragment was excavated in 2012 during ongoing archaeological work at Denisova Cave, part of the East Gallery, one of several distinct chambers within the wider cave system that researchers have excavated separately over decades. At the moment it came out of the ground, it was simply one piece of bone among an enormous quantity of material recovered from that season's dig. Archaeological excavation at a site with this much history produces staggering volumes of material, most of it animal bone, fragmented by time, by the weight of accumulated sediment, by scavengers who may have gnawed at remains long before any human returned to bury or move them. Sorting that material by species, let alone by whether any given fragment might belong to an ancient human rather than an ancient animal, is not something that can be done reliably by eye alone, especially when a fragment is as small and nondescript as this one turned out to be. For 4 years, the fragment sat uncategorized, one among roughly 2,000 similarly unidentified bone pieces collected from various excavations at the site. This delay was not negligence. It reflects an honest and important truth about archaeology that rarely makes it into popular retellings of major discoveries. The vast majority of what comes out of the ground at any major excavation cannot be immediately understood. Identification takes time, funding, specialized expertise, and often new technology that simply did not exist at the moment of excavation. Denisova Cave had already produced discoveries significant enough to draw international attention, most notably the 2010 identification of the Denisovans themselves from a single finger bone. That success meant researchers had every reason to keep looking closely at unidentified fragments from the same site. But even with that motivation, the sheer volume of material meant progress was gradual. The breakthrough that finally sorted human from animal within that tray of fragments came from a technique called zooarchaeology by mass spectrometry, generally referred to by researchers using the shorthand ZOO-MS. The method works by extracting collagen, the primary structural protein in bone, and analyzing its molecular composition using mass spectrometry, a technique that measures the precise mass of molecular fragments to identify what they are made of. Collagen structure varies slightly, but measurably between different animal species and between animals and humans. By running fragments through this analysis, researchers can sort bone by broad biological category without needing enough visible anatomical detail to identify a fragment by eye, which is exactly the problem that had kept 2,000 unidentified pieces sitting in storage for years. A research team that included Samantha Brown, working alongside colleagues including archaeologist Tom Hyam of Oxford University, applied this technique to the tray of unidentified Denisova Cave fragments. Among the results, one small piece stood out. Its collagen profile matched not an animal but an archaic human. That single result transformed an anonymous splinter into a priority sample, worthy of the kind of detailed genetic analysis that only a handful of laboratories in the world were equipped to perform. The fragment was sent to the Max Planck Institute for Evolutionary Anthropology in Leipzig, Germany, a laboratory that had, by that point, become the global center for ancient human genetic research, largely due to the pioneering work of geneticist Svante Pääbo, who had spent decades developing methods to extract usable genetic material from bones and teeth that were, in many cases, tens of thousands or even hundreds of thousands of years old. Ancient DNA is notoriously difficult to work with. It degrades over time, breaking into short, damaged fragments. It becomes contaminated by the DNA of bacteria, fungi, and other organisms that colonize buried remains over millennia. It becomes contaminated too by the DNA of every researcher, excavator, and laboratory technician who has ever touched the sample, however carefully. Extracting a clean, reliable genetic signal from something like a 2.5 centimeter bone fragment, buried for tens of thousands of years, is closer to forensic reconstruction than simple laboratory testing. It requires clean rooms, strict protocols, and repeated verification to separate genuine ancient signal from modern contamination. The genetic analysis was led by Viviane Slon, a member of Pääbo's research group, who focused specifically on the sequencing and interpretation of the sample. Initial results confirmed what the ZooMS screening had already suggested. The fragment belonged to an ancient human. Mitochondrial DNA analysis, which traces genetic material passed down exclusively through the maternal line, gave researchers their first specific clue about the fragment's identity. It matched Neanderthal mitochondrial DNA, telling them, with a fair degree of confidence, that whoever this fragment belonged to had a Neanderthal mother, or a Neanderthal maternal lineage stretching back through her mother's side of the family. That alone would have been a meaningful, if not especially unusual, result. Neanderthal remains, while rare, are not unprecedented, and this would have simply been added to the broader catalogue of known Neanderthal individuals. But mitochondrial DNA tells only part of the story. It traces one narrow genetic thread, the maternal line, and says nothing directly about the rest of an individual's ancestry. To understand the fuller picture, researchers needed to sequence the nuclear genome, the much larger and more complete set of genetic material inherited from both parents, carried in the nucleus of every cell. That deeper analysis is where the story changed entirely. When Slon's team examined the nuclear genome extracted from the fragment, they found something that did not fit the simple picture the mitochondrial result had suggested. Roughly half of the individual's genetic material matched Neanderthal ancestry, consistent with the maternal signal already found. But the other half did not match Neanderthal ancestry at all. It matched Denisovan ancestry, and it did so with a clarity and consistency that ruled out the possibility of a distant, diluted mixture. This was not a genome carrying a small percentage of Denisovan heritage inherited many generations back, the kind of trace signal researchers had already documented in some modern human populations. This was a near-perfect 50-50 split, the unmistakable genetic signature of a child born to one parent from each population. The team ran the analysis multiple times, checking and rechecking the result, aware of exactly how extraordinary a claim they were about to publish. Contamination had to be ruled out. The possibility of a simple mixing of two separate ancient samples, accidentally combined during excavation or laboratory processing, had to be ruled out as well. Every check confirmed the same conclusion. This was one individual, one person, one genome carrying the direct, unmixed genetic contribution of two parents from two different, deeply diverged human populations. Further analysis allowed the team to determine additional details about the individual. Examination of specific regions of the genome linked to sex determination confirmed the individual was female. Analysis of bone density and cortical thickness, techniques used to estimate age at death from skeletal fragments, even when only a small piece survives, suggested she was at least 13 years old when she died, likely in her early teenage years. She had a name now, at least among the scientists who studied her, not a name from her own language, lost along with every word her people ever spoke. A working name, chosen the way researchers often choose names for significant specimens, drawing on the site of discovery and the affectionate shorthand that develops among people who spend years studying the same small fragment of bone. They called her Denny. Chapter 5. What a Body Remembers. A genome is, in a very real sense, a kind of memory. Not the memory of a single life, but a compressed chemical record of every ancestor who ever contributed to the body carrying it. Understanding what Denny's genome actually told researchers and how they read it with confidence rather than guesswork means understanding a little about how DNA carries the fingerprints of a family tree, even one stretching back hundreds of thousands of years. Every person inherits half of their genetic material from their mother and half from their father. That half-and-half split happens at conception and never again in quite the same clean way. A child born to two parents from the same broadly similar population will, over many generations, carry a genome that reflects a long, blended mixture of countless ancestors, each contributing smaller and smaller individual fractions as the generations pass. By the time you look at someone whose ancestors have been part of the same general population for hundreds of generations, their genome reads as a smooth, well-mixed average of that population's genetic diversity. There is no obvious seam, no visible line where one ancestor's contribution ends and another's begins. That is emphatically not what Sloan's team found when they analysed Denisova 11's genome. Instead of a smooth, blended signal, they found something closer to two distinct genetic sources sitting side by side, cleanly divided, almost like two different colours of paint that had been poured into the same container but had not yet had time to mix. Roughly half of her genetic material aligned closely and consistently with known Neanderthal genomes. The other half aligned just as closely and consistently with known Denisovan genomes. There was no gradual blending pattern, no evidence of many prior generations of mixing softening the boundary between the two ancestries. The division was sharp, immediate, and unmistakable. This pattern is the genetic signature of what scientists call an F1 hybrid, a term borrowed from genetics more broadly, referring specifically to the first-generation offspring of two genetically distinct parent populations. An F1 hybrid inherits one complete, unmixed set of chromosomes from each parent, meaning that the two ancestries sit side by side in the child's genome without having had any opportunity to blend, because blending only begins to occur in later generations, as descendants inherit shuffled combinations from parents who are themselves already mixed. Denny's genome showed no signs of that later shuffling. It showed the clean, immediate signature of a child whose mother and father belonged, in that moment, to two populations that had not previously mixed within her own direct family line. To confirm this interpretation and to rule out other possible explanations, researchers compared Denny's genome against reference genomes from other known Neanderthal individuals and other known Denisovan individuals drawn from previous discoveries at the same cave and elsewhere. This comparison allowed them to check whether her Neanderthal-matching genetic material behaved consistently with genuine Neanderthal ancestry, and whether her Denisovan-matching material behaved consistently with genuine Denisovan ancestry, across many different points along her genome. It did, in both cases, with remarkable consistency. There was no reasonable alternative explanation left standing. This was not sample contamination. This was not an accidental mixture of two separate specimens during handling. This was one individual whose two halves of ancestry came from two different, deeply diverged human populations, combined in exactly the way biology combines the genetic material of two parents into one child. Once the basic finding was confirmed, researchers pushed further, using the genome to extract even more specific information about each of Deni's parents individually, despite having no physical remains from either one. The maternal side of her ancestry, drawn from the Neanderthal half of her genome, was compared against other known Neanderthal genomes from different sites across Eurasia. This comparison revealed the detail mentioned earlier, that her mother's ancestry was more closely related to Neanderthal populations that later lived in Western Europe than to an earlier Neanderthal individual whose remains had also been recovered from Denisova Cave itself, an individual who lived considerably earlier in the cave's long history. This finding suggested that Deni's maternal lineage represented a distinct, likely more recent wave of Neanderthal migration into the region, separate from whatever earlier Neanderthal population had also, at some point, called the same cave home. In other words, more than one distinct Neanderthal population passed through this valley across the span of the cave's occupation, and Deni's mother belonged to a later group than the very first. The paternal side of her ancestry, drawn from the Denisovan half of her genome, produced its own surprise. Researchers compared her Denisovan matching genetic material against the genome of another individual known as Denisova 3, the original finger bone specimen that had first identified the Denisovan population back in 2010, and against a second individual known as Denisova 4. What they found was that Deni's father's ancestry was more closely related to a later Denisovan individual found in the cave, known as Denisova 2, than to the earlier specimens, again suggesting the presence of more than one distinct Denisovan population moving through the region across different periods. And then came the detail that added an entire extra layer to the story. When researchers examined her father's genetic contribution more closely, they found faint but detectable traces of Neanderthal ancestry within it, distinct from the Neanderthal ancestry inherited from Deni's mother. This trace was old, degraded by many intervening generations, consistent with an admixture event that had occurred hundreds of generations before her father's own birth, estimated by researchers to be somewhere in the range of several hundred generations earlier. In practical terms, this meant that Deni's father, himself fully Denisovan in the sense that both of his own parents belonged to that population, nonetheless carried within his own genome the distant genetic legacy of an earlier meeting between Denisovans and Neanderthals, an encounter that had happened long before he was born, likely in an entirely different time, and possibly an entirely different place. This detail transformed the story from a single, isolated meeting into something with much deeper roots. Deni's own existence proved that her mother and father had come together at least once, resulting in her birth. But her father's genome proved that this was not the first time, and likely not the only time, that Neanderthals and Denisovans had crossed paths and produced children together. Somewhere, centuries before Deni's own parents ever met, an earlier Neanderthal and an earlier Denisovan had done the same thing, and that earlier union's genetic legacy had quietly persisted, diluted but present, through hundreds of generations of purely Denisovan descendants, until it resurfaced, so to speak, in the DNA of a man whose own daughter would go on to prove that these two populations had never really stopped finding each other. For researchers studying ancient human populations, this was a genuinely significant shift in understanding. A single hybrid individual could, in principle, be dismissed as an extraordinary but essentially isolated event, a rare biological accident with limited broader significance. Evidence of repeated, separate instances of interbreeding across different generations pointed towards something else entirely. It suggested that wherever and whenever these two populations found themselves in proximity, whether through shared territory, competing claims to valuable shelter like Denisova Cave, or simple chance encounters during seasonal movement, interbreeding was not a rare, isolated aberration. It was something that happened again and again, whenever the opportunity presented itself. Chapter 6. Not the only ones. Denny's discovery did not exist in isolation. It arrived as the most dramatic piece of evidence within a much broader, decades-long scientific effort to understand just how much interbreeding occurred between modern humans, Neanderthals, and Denisovans throughout the deep past. And the picture that emerged from that broader effort, once researchers had time to fully absorb it, was that ancient humanity was far more interconnected than almost anyone had assumed even 20 years earlier. The first solid evidence that Neanderthals and modern humans had interbred came from a landmark study published in 2010, the same year the Denisovans themselves were first identified. Researchers sequencing the Neanderthal genome for the first time compared it against the genomes of modern humans from populations around the world and found something unexpected. People with ancestry outside of Africa, meaning the majority of the world's population whose ancestors migrated out of the African continent tens of thousands of years ago, carried a small but consistent percentage of Neanderthal DNA in their genomes, typically somewhere between 1 and 2%. People with ancestry entirely within sub-Saharan Africa, whose lineage had never passed through the regions where Neanderthals lived, generally carried little to none. The explanation was straightforward once the pattern became clear. At some point after early modern humans began migrating out of Africa, likely somewhere in the Near East, they encountered Neanderthal populations who were already established across that region. The two groups interbred, and the genetic legacy of that mixing was carried forward by the descendants of those unions, spreading gradually through the growing modern human population as it continued to migrate and expand across Europe and Asia. Every non-African person alive today, whether they know it or not, carries a small physical inheritance from that ancient meeting, scattered through their genome in fragments, the accumulated echo of relationships that took place tens of thousands of years ago between two different kinds of human beings. A similar pattern, though geographically distinct, emerged for Denisovan ancestry. Researchers found that certain modern human populations, particularly in Melanesia, Papua New Guinea, and parts of Australia and Southeast Asia, carry a notably higher percentage of Denisovan ancestry than populations elsewhere in the world, in some cases reaching 4 to 6% of their total genome. This pattern suggested that as early modern human populations continued their migration eastward and southward through Asia, at some point encountering Denisovan populations along the way, further interbreeding occurred, this time leaving its genetic mark specifically on the ancestors of people who would eventually settle the islands and continent of the Australasian region. Genetic estimates place at least one significant period of Denisovan and modern human interbreeding at approximately 46,000 years ago, though the full picture likely involves more than one distinct episode of contact across different times and places, given how variable Denisovan ancestry levels are across different modern populations. What makes this picture even more intricate is evidence that Neanderthals and Denisovans themselves interbred with each other on more than one occasion, as Denny's own existence and her father's own trace Neanderthal ancestry both independently demonstrate. Taken together, all of this evidence points toward a deep past that looked less like three separate, isolated species living apart from one another, occasionally bumping into each other by unfortunate accident, and more like a loosely connected web of related human populations, distinct from one another in many ways, shaped by different environments and different histories, but never fully separated by an impassable biological wall. Wherever they met, across tens of thousands of years and thousands of kilometers, they had children. This broader pattern changes how researchers now think about what actually happened to Neanderthals and Denisovans as distinct populations. The old, simple narrative held that these were separate species that went extinct, replaced entirely by modern humans who arrived, competed for the same resources, and eventually won that competition, leaving Neanderthals and Denisovans to disappear without a genetic trace. The evidence gathered over the past 15 years complicates that narrative considerably. Rather than a clean replacement, with one population vanishing entirely while another moved in, the more accurate picture appears to be one of gradual absorption. As modern human populations expanded into regions already inhabited by Neanderthals and Denisovans, interbreeding meant that at least some portion of those older populations' genetic material did not vanish. It was folded into the growing modern human population instead, carried forward generation after generation, diluted over time but never fully erased. This does not mean Neanderthals and Denisovans, as distinct, self-sustaining populations with their own separate cultures and ways of life, did not eventually disappear. The physical, cultural, and behavioral evidence associated with these populations does fade from the archaeological record over time, and by roughly 40,000 years ago, distinct Neanderthal sites across Europe largely stop appearing, while Denisovan sites become increasingly rare in the millennia that follow. Something did happen that brought an end to these populations existing as separate, independent groups. What researchers debate, and continue to research actively, is the relative role different factors played in that ending, including competition for resources with expanding modern human populations, environmental and climate pressures, small population sizes making these groups more vulnerable to random extinction events, and gradual absorption through ongoing interbreeding with modern humans, which would have reduced their genetic and cultural distinctiveness over successive generations even without a single dramatic cause of disappearance. What is not in doubt, because it survives directly in the bodies of billions of people alive today, is that these populations did not vanish without a trace. Every time a modern human, anywhere in the world today, whose ancestry runs even partly outside sub-Saharan Africa, undergoes a genetic test and finds a small percentage of Neanderthal or Denisovan ancestry listed in their results, they are looking at a direct, measurable legacy of encounters like the one that produced Deni, encounters that took place not once but likely many times, across tens of thousands of years, between ancient human populations that modern imagination once assumed to be entirely separate. Deni's significance within this broader story is that she represents the clearest, most direct, least ambiguous piece of evidence anyone has ever found for this pattern of interbreeding. Modern human genomes, carrying small percentages of Neanderthal or Denisovan ancestry, are the diluted, distant echo of ancient unions, filtered and reshuffled through thousands of subsequent generations. Deni is not an echo. She is the union itself, preserved directly in bone, a single, undeniable, first-generation proof that two different kinds of ancient humanity did not simply cross paths from a safe genetic distance. They came close enough, often enough, in enough different times and places, to become, quite literally, family. Chapter 7. What her bone could not answer. For all the certainty that Deni's genome provided about her ancestry, the physical evidence of her life remains almost painfully thin, and it is worth being honest about exactly how much and how little a single bone fragment can tell us about a person. The specimen recovered from Denisova Cave, catalogued as Denisova 11, consists of a single piece of long bone, roughly 2.5 centimeters in length, believed to come from either an arm or a leg, though researchers have not been able to determine with full certainty which limb or which specific bone it originally belonged to. This is not a failure of the research team. It is simply the reality of working with a fragment this small and this incomplete. There is no skull to examine for facial structure, no teeth to study for wear patterns or diet, no pelvis to confirm sex through traditional skeletal analysis, though genetic analysis was able to establish this instead. No spine, no ribs, no hands, no feet, just a small piece of one limb bone, denser and more durable than many other bones in the human body, which is likely part of why it survived at all across 90,000 years, while so much else around it did not. From this single fragment, researchers extracted an extraordinary amount of information, but it is worth cataloguing clearly what that information could and could not tell them. Genetic analysis confirmed her sex as female, confirmed her mixed Neanderthal and Denisovan ancestry, and provided enough detail to reconstruct aspects of both parents' broader population histories. Analysis of the bone's cortical thickness, essentially how dense and mature the bone tissue had become by the time of death, allowed researchers to estimate her age. Cortical bone continues to thicken and mature throughout childhood and adolescence, following patterns that are reasonably well understood from studies of skeletal development. Based on this analysis, researchers concluded she was at least 13 years old at the time of her death, though the exact upper limit of her age remains less certain, given that only this one fragment survives. What the bone cannot tell us, and what no amount of genetic or structural analysis will ever recover, is anything about how she died, what her daily life looked like, what her relationships with her family and community were like, or what, if anything, made her particularly memorable to the people who knew her. We do not know if she was sick for a long period before her death, or if her death was sudden. We do not know if her body was deliberately buried with any care or ritual, or whether the fragment ended up in the cave sediment through some other process entirely, perhaps carried there by scavengers, or displaced through natural sediment movement over the many millennia since her death. The layer of sediment in which the fragment was found does not, on its own, provide the kind of detailed archaeological context that would allow researchers to answer these questions with confidence. This gap between what genetics can prove and what archaeology can reconstruct is one of the most important, if less dramatic, lessons embedded in Deni's story. Modern genetic technology has become extraordinarily powerful, capable of extracting detailed ancestry information from bone fragments so small and so degraded that earlier generations of scientists would never have imagined they held any useful information at all. But that power does not extend to recovering the texture of an individual life. Genetics can tell you who someone's parents were. It cannot tell you whether that person was loved, or how they spent their days, or what frightened them, or what made them laugh, assuming laughter, in whatever form it took, was part of the emotional world of a 13-year-old girl living in Siberia 90,000 years ago. Those details, if they ever existed in any recoverable form, are gone, and responsible science does not pretend otherwise by inventing details to fill the silence. What researchers can do, responsibly, is use the broader archaeological and genetic evidence from Denisova Cave and from the wider study of Neanderthal and Denisovan populations to construct an informed, honest picture of the kind of world Deni likely lived in, without claiming false certainty about details specific to her individual experience. This is the difference between responsible historical reconstruction and speculation dressed up as fact, and it matters enormously in a story like this one, where the temptation to fill silence with invented detail is strong precisely because the real story is already so compelling. Based on broader evidence from Neanderthal and Denisovan sites, including Denisova Cave itself, researchers have a reasonably good general understanding of the kind of environment a 13-year-old girl of mixed ancestry would likely have grown up in during this period. The Altai region, roughly 90,000 years ago, experienced a climate that fluctuated between cooler and somewhat milder periods, part of the broader pattern of climatic shifts that characterized the late Pleistocene across Eurasia. Isotope analysis and pollen evidence recovered from cave sediments at Denisova and similar sites in the region suggest a mixed environment of forest and steppe, providing access to a range of large game animals, including species such as bison, horses, and deer, alongside smaller mammals and, seasonally, fish from nearby rivers. Both Neanderthal and Denisovan populations in this broader region appear, based on tool evidence and animal remains found alongside their fossils, to have practiced a mobile hunting and gathering lifestyle, likely moving across a home range that shifted with the seasons, following game and taking advantage of different resources at different times of year, while returning periodically to reliable, sheltered locations like caves for extended stays, particularly during the harshest parts of winter. A child growing up in this world would have been raised within a small, close-knit group, likely a band numbering anywhere from a handful of individuals to a few dozen, given what is understood more broadly about the likely small population sizes and social groupings of both Neanderthal and Denisovan populations during this period. This matters for understanding the circumstances of Deni's own existence. In a world of small, dispersed populations, encounters between different groups, even different populations, would not have been common daily occurrences. They would have been comparatively rare events, shaped by shared use of valuable limited resources like caves and river valleys, or by the periodic overlap of seasonal movement ranges. The fact that Deni's parents met at all, given how relatively small and scattered both Neanderthal and Denisovan populations likely were across this vast landscape, is itself a meaningful piece of the puzzle. It suggests that whatever territorial or seasonal patterns brought different groups into contact around places like Denisova Cave, they were consistent enough and happened often enough across generations for such meetings and the relationships that sometimes followed them to occur more than once, as her father's own trace Neanderthal ancestry independently confirms. There is also a genuinely important detail buried within the technical analysis of Deni's genome, one that researchers were careful to raise honestly rather than gloss over. It remains unknown whether hybrid individuals like Deni were biologically fertile, capable of having children of their own, who could carry forward both Neanderthal and Denisovan ancestry into future generations. Some hybrid animals in the natural world are fertile, capable of passing on genes to further descendants. Others are not, or are only partially fertile, facing reduced reproductive success compared to non-hybrid individuals. Because Deni died as a young teenager, before she would likely have had children of her own, her individual case cannot answer this question directly. What can be said is that the broader genetic evidence, showing traces of repeated interbreeding across multiple generations, reflected in both Neanderthal and modern human genomes, and in the trace Neanderthal ancestry found within Deni's own Denisovan father, strongly suggests that at least some hybrid individuals across the broader population were able to reproduce successfully, carrying mixed ancestry forward into subsequent generations, even if we cannot say this with absolute certainty about Deni specifically. Chapter 8. The Ones Who Came Before and After. Her Deni's story would be extraordinary, even if she stood entirely alone in the archaeological record. A single, isolated case of interbreeding never repeated before or after. But the deeper researchers have looked, both at Denisova Cave itself and at sites across Eurasia, the clearer it has become that she was neither the first nor the last piece of evidence pointing toward a long, complicated pattern of contact between these ancient populations. Denisova Cave itself has produced a remarkable roster of individual archaic humans across the decades of excavation there, each one adding another piece to an increasingly intricate picture. The very first Denisovan individual identified, back in 2010, came from a finger bone catalogued as Denisova 3, recovered from a layer of sediment that genetic dating techniques place at somewhere between 50 and 76,000 years old, making that individual younger than Deni herself. A second specimen, a large molar tooth catalogued as Denisova 4, provided further genetic confirmation of the Denisovan population and helped researchers understand more about their physical characteristics, given how unusually large and robust Denisovan teeth appear to have been compared to those of modern humans or Neanderthals. Later excavations recovered additional specimens, including another tooth and a small fragment of skull, each one adding incrementally to the genetic and physical picture of this still mysterious population. The cave has also produced Neanderthal remains distinct from the mixed ancestry found in Deni herself, including an individual whose genome researchers were able to compare directly against Deni's own Neanderthal ancestry, revealing, as discussed earlier, that this earlier individual represented a different, likely earlier wave of Neanderthal presence in the region than the population Deni's own mother belonged to. This detail alone tells us that the cave was not occupied by a single, unchanging Neanderthal population across its entire history, but by at least two distinct Neanderthal groups, arriving in the region at different points, separated by enough time and enough genetic distance for researchers to detect the difference clearly within the genetic record. Beyond the individual specimens found directly within Denisova Cave, broader genetic surveys of sediment from the site, examining ancient DNA preserved not in bone but in the dirt itself, a technique that has become increasingly important in archaeological research over the past decade, have confirmed the presence of both Neanderthal and Denisovan genetic material across multiple, distinct layers spanning enormous stretches of time. This sediment-based evidence extends the known window of both populations' presence at the site, considerably beyond what could be established from the limited number of actual bone fragments recovered, essentially confirming that both groups returned to this same location repeatedly across many thousands of years, long enough for their populations to shift, change, and in Deni's case, occasionally combine. Beyond Siberia, evidence for Denisovan presence and their genetic legacy continues to expand as research progresses. The 2019 identification of the Shahi mandible, the jawbone discovered decades earlier by a monk in Baishia Karst Cave on the Tibetan Plateau, extended the known Denisovan range by thousands of kilometers, proving that this population was not confined to a single mountain valley in Siberia, but had spread across an enormous stretch of Asia, adapting successfully to environments as different as the temperate forests near Denisova Cave and the extreme high-altitude conditions of the Tibetan Plateau. Subsequent sediment DNA analysis at that same Tibetan site confirmed Denisovan genetic material present across multiple layers, spanning roughly 100,000 years down to as recently as 45,000 years ago, suggesting Denisovans persisted in that challenging environment for an extraordinarily long stretch of time, potentially overlapping with the arrival of modern humans into the region. More recent archaeological work at Baishia Karst Cave has continued to add detail to the picture of how Denisovans actually lived day to day. Excavations examining animal remains recovered from the site have identified evidence of sustained hunting and butchery of a range of species adapted to high-altitude environments, including blue sheep, marmots, and various birds, alongside evidence suggesting the population processed and likely stored food resources across different seasons, a level of planning and environmental adaptation that speaks to genuine sophistication rather than marginal precarious survival. The wider genetic legacy of Denisovan ancestry in modern human populations has also continued to reveal new layers of complexity as research has progressed. Beyond the well-documented elevated levels of Denisovan ancestry in Melanesian and some Southeast Asian and Australian populations, researchers have identified evidence suggesting that modern human ancestors may have interbred with more than one distinct Denisovan population at different points and in different regions, each contributing slightly different genetic signatures to different modern populations. This would mean that Denisovans themselves were not a single uniform group occupying one unbroken territory, but a more genetically diverse population spread across a wide range of environments across Asia, echoing the same kind of internal diversity researchers have documented within Neanderthal populations across Europe and Western Asia. Perhaps the most striking modern legacy of Denisovan ancestry involves a specific genetic variant found in the gene EPAS1, which plays a critical role in how the human body regulates its response to low oxygen levels. Modern Tibetan populations, who live and work successfully at altitudes that would cause serious altitude sickness in most other human populations, carry a version of this gene that researchers have traced directly back to Denisovan ancestry. This variant appears to have entered the modern human gene pool through ancient interbreeding with Denisovan populations, and it provides modern Tibetan populations with a measurable, life-preserving advantage in an extreme environment, allowing more efficient oxygen use without the excessive increase in red blood cell production that can cause serious long-term health complications in other populations attempting to live at extreme altitude. This single genetic detail carries enormous weight when placed alongside Deni's own story. It means that the population her father belonged to, a population known to us today through only a small handful of fragmentary remains, a population so poorly understood that researchers still cannot say with full confidence what they looked like, left behind a genetic gift so valuable that it continues, at this very moment, to help millions of people breathe more easily in one of the most extreme environments on Earth. The Denisovans did not simply vanish into the silence of prehistory. Pieces of them are still here, still functioning, still keeping people alive, embedded quietly within the genomes of populations who likely have no idea, walking through their daily lives at high altitude, that their bodies carry a survival trait inherited from a population that stopped existing as a distinct group tens of thousands of years ago. Deni herself did not live long enough to pass anything forward. Her own genetic contribution to the future ended with her death as a young teenager. But the broader pattern her existence helped confirm, that Neanderthals and Denisovans interbred repeatedly, across generations, wherever their populations came into contact, means that somewhere in the deep, tangled roots of human ancestry, individuals like her, hybrid children born of two different kinds of ancient humanity, may have been far more common than the fossil record alone will ever be able to prove. She is not necessarily unique in what she was. She is simply, so far, the only one science has managed to prove existed, pulled by chance from a tray of 2,000 anonymous fragments, identified through a combination of careful excavation, patient laboratory work, and a molecular technique that did not even exist when her bone was first placed in the ground. The tools that made the discovery possible. It is worth pausing, before moving further into the wider implications of Deni's discovery, to understand exactly how remarkable the underlying science actually was, because the story of the technology behind this find is, in its own way, as extraordinary as the discovery itself. Ancient DNA research did not always exist as a reliable scientific field. For decades, the idea of extracting usable genetic material from bones tens of thousands of years old was treated with deep skepticism within the broader scientific community, and for good reason. Early attempts at ancient DNA extraction, dating back to the 1980s and 1990s, were plagued by contamination problems so severe that entire published findings were later retracted or discredited, because researchers had unknowingly sequenced modern bacterial DNA or even their own genetic material, rather than anything genuinely ancient. It took years of methodological development, painstaking protocol refinement, and the construction of specialized cleanroom laboratories designed specifically to minimize any possible contamination from modern DNA sources before ancient DNA research became a field capable of producing results that the broader scientific community could trust. Svante Pääbo, the researcher whose laboratory eventually sequenced Denis' genome, spent much of his career at the center of this methodological transformation. His work, alongside colleagues across multiple institutions, gradually developed the techniques that made it possible to extract short, degraded fragments of genetic material from bones and teeth, piece them back together using sophisticated computational methods, and distinguish genuine ancient signal from the noise of contamination and degradation. This work culminated in 2010 in the first complete sequencing of a Neanderthal genome, a landmark achievement that itself took years of dedicated effort and provided the essential comparison baseline that would later allow researchers to identify Denis' mixed ancestry with confidence. Without that earlier achievement, sequencing Denis' genome and correctly interpreting what it showed would simply not have been possible. Her discovery stood on the shoulders of over a decade of prior, less publicly celebrated scientific groundwork. The specific technique that first flagged Denis' fragment as human rather than animal, zooarchaeology by mass spectrometry, represents its own quiet technological triumph. Developed originally for other applications in archaeological science, the method allows researchers to identify the species origin of bone fragments using collagen peptide analysis, a process that requires only a tiny sample, often just a few milligrams of bone powder, extracted in a way that causes minimal damage to the specimen itself. This matters enormously when working with irreplaceable, rare archaeological material, where destructive testing methods that require larger samples would risk damaging or entirely consuming a specimen before its full scientific value could be extracted. The relatively non-destructive nature of ZooMS screening meant that Denis' fragment could be tested, identified as human, and then preserved well enough for the subsequent, more detailed genetic analysis that would ultimately reveal her extraordinary ancestry. Once a fragment has been genetically sequenced, interpreting the results requires its own layer of sophisticated computational work. Researchers do not simply read a genome the way one might read a book. Ancient DNA typically survives only in short, damaged fragments, often just a few dozen genetic letters long, chemically altered by tens of thousands of years of degradation in ways that leave distinctive damage patterns researchers have learned to recognize and account for. Reconstructing a usable genome from millions of these short fragments requires powerful computational tools capable of aligning fragments against reference genomes, identifying where each piece belongs, and statistically accounting for the various forms of damage and uncertainty inherent in working with material this old. The fact that researchers could confidently distinguish, within Denis' genome, not only her overall mixed ancestry, but the more specific detail of exactly which populations her mother's and father's lineages most closely resembled, down to identifying which other individual specimens from the same cave they were more or less related to, reflects a level of analytical precision that would have been entirely unimaginable to researchers working in this field even 2 decades earlier. This technological context matters because it explains why a discovery like Denis' happened when it did, rather than earlier or later. The bone fragment itself had been sitting in the ground, and later in a laboratory drawer for a span of time that dwarfs the entire history of modern science many times over. What changed was not the fragment. What changed was humanity's own technological capability to finally read what that fragment had been quietly holding all along. In a very real sense, Denis' discovery is not only a story about ancient human history, it is also a story about the extraordinary, decades-long scientific effort required to build the tools capable of recovering that history in the first place. An effort involving hundreds of researchers across multiple countries, working in specialized clean room laboratories, developing statistical methods, refining extraction protocols, and slowly, patiently, building the foundation that made a discovery like hers possible. What survival actually looked like. To understand the world Denis was born into with real honesty, rather than through vague, generic gestures toward prehistoric hardship, it helps to look closely at what the archaeological evidence actually tells researchers about daily survival for populations like hers, in a region and climate as demanding as the Altai Mountains during this period of the late Pleistocene. Winters in this region would have been long and severe, with temperatures capable of dropping to levels that make survival for an unprotected human body a matter of hours rather than days. Both Neanderthal and Denisovan populations left behind clear evidence of adaptation to exactly this kind of challenge. Hearths recovered from multiple layers within Denisova Cave show evidence of sustained, controlled fire use, not simply occasional or opportunistic burning, but organized fire management sophisticated enough to provide reliable heat and light across extended periods of occupation. Fire in this context was not a convenience. It was the difference between a livable shelter and a death trap, and the consistency of hearth evidence across so many layers of the cave's long occupation history suggests that both populations understood and depended on fire management as a core survival skill, passed down and practiced across many successive generations. Clothing, though it leaves far less direct archaeological evidence than stone tools or hearths, given how rarely organic material like leather or fur survives across tens of thousands of years, can be inferred indirectly through other evidence. Bone tools identified at various Neanderthal sites across Eurasia, including specialized implements interpreted by researchers as likely used for working hides, suggest that at least some Neanderthal populations processed animal skins into usable material, very likely for clothing suited to cold climate survival. Given that Denisovans occupied environments ranging from the cold valleys of southern Siberia to the even more extreme high-altitude conditions of the Tibetan Plateau, it is reasonable to conclude that some form of effective cold-weather clothing was a necessary and likely well-developed part of Denisovan material culture as well, even though direct physical evidence of Denisovan clothing itself has not survived. Food acquisition in this environment required sophisticated, coordinated effort. Animal remains recovered from Denisova Cave across its various occupation layers include a range of species, from large game like bison, horses, and various deer species, down to smaller mammals, and based on evidence from certain layers, fish taken from the nearby river system. Successfully hunting large, dangerous game animals using the stone-tipped spears and other weapons available to populations during this period required genuine skill, patience, and very likely coordinated group effort, given the risks involved in approaching close enough to a large animal to deliver an effective, close-range strike with the weapons technology available at the time. This was not opportunistic, haphazard survival. This was skilled, practiced, socially coordinated work, requiring the kind of accumulated knowledge that gets passed down deliberately from experienced hunters to younger members of a community, generation after generation. Beyond hunting, Evidence from dental analysis and chemical residue studies conducted on Neanderthal remains from various sites across Eurasia has revealed a far more varied diet than the meat-heavy stereotype long assumed by earlier generations of researchers. Starch granules preserved in dental calculus, the hardened plaque that builds up on teeth over a lifetime and can survive remarkably well across tens of thousands of years, have been identified on Neanderthal teeth from multiple sites, showing evidence of plant consumption, including various roots, seeds, and in some cases, plants that appear to have been cooked before eating, based on visible changes to the starch structure consistent with heat processing. This evidence paints a picture of a population with a genuinely varied, seasonally adapted diet, drawing on whatever combination of hunted meat and gathered plant material was available across different times of year, rather than a population surviving on meat alone. Social structure during this period, though impossible to reconstruct with the kind of specific detail available for better documented historical societies, can be reasonably inferred from a combination of comparative evidence and direct archaeological indicators. Group sizes for both Neanderthal and Denisovan populations were very likely small by modern standards, probably ranging from a handful of closely related individuals up to a few dozen people forming a broader, loosely connected band, based on comparisons with the general population dynamics documented among later hunter-gatherer societies, and on the physical capacity of known Neanderthal and Denisovan shelter sites to comfortably support only a limited number of people at any given time. Within groups of this size, survival almost certainly depended on close cooperation, shared labor, and the kind of accumulated collective knowledge about which plants were safe to eat, which locations offered reliable water and shelter, which seasonal patterns of animal movement could be predicted and exploited, that would have been passed down carefully from older, more experienced members of the community to younger ones across many years of direct teaching and practical experience. It is within this world, demanding, skilled, cooperative, and shaped by constant close attention to the practical realities of survival in a harsh environment, that Deni would have been born and would have spent whatever portion of her short life the evidence allows us to imagine, even as the specific, individual details of her own daily experience remain, and will likely always remain, beyond what the physical evidence can recover. The wider search that continues. Deni's discovery did not close the book on the study of ancient human interbreeding. If anything, it opened an entirely new chapter of active, ongoing research, one that continues to develop with each passing year as new techniques emerge and new sites are excavated across Asia and beyond. In the years since 2018, researchers have continued applying the same combination of zoo-MS screening and detailed genetic sequencing to unidentified bone fragments recovered from Denisova Cave and other significant sites across Eurasia, hoping to identify additional hybrid individuals or additional evidence that might further clarify the pattern of contact between Neanderthals, Denisovans, and modern humans. While no second individual has yet been found matching Deni's remarkable first-generation hybrid status, ongoing sediment DNA analysis, the technique of extracting genetic material directly from cave floor dirt rather than from identifiable bone fragments, has continued to expand researchers' understanding of when and where both populations were present across various sites, gradually filling in a more detailed picture of their overlapping ranges across both time and geography. Researchers have also continued refining their understanding of the broader genetic legacy left behind by both populations within modern human genomes. Advances in genetic sequencing technology and analytical methods have allowed scientists to identify increasingly specific genetic variants inherited from Neanderthal or Denisovan ancestors that continue to influence traits in modern human populations today, extending well beyond the well-known example of high-altitude adaptation in Tibetan populations. Research has identified Neanderthal inherited genetic variants linked to aspects of immune system function, skin and hair characteristics, and, in some studies, even certain patterns of sleep and mood regulation. Though researchers are careful to note that establishing a clear genetic inheritance is quite different from establishing a simple, direct causal relationship between an ancient variant and a specific modern trait, given how many other genetic and environmental factors typically interact to shape any given human characteristic. This ongoing research continues to reveal just how deeply and specifically ancient interbreeding shaped the genetic makeup of people alive today, extending the significance of discoveries like Denis well beyond the specific ancient context in which she lived. The broader search for additional Denisovan remains, in particular, remains one of the most active areas of paleoanthropological research today, given how strikingly limited the physical fossil evidence for this population still is, compared to the extensive genetic evidence for their impact on modern human populations. Researchers continue to excavate at Denisova Cave itself, working through remaining unexcavated sediment layers that may yet contain additional Denisovan or Neanderthal remains. Similar excavation work continues at Baishia Karst Cave on the Tibetan Plateau, and researchers have begun exploring other cave systems across China and broader East Asia, hoping to identify additional sites that might have hosted Denisovan populations, given genetic evidence suggesting their range likely extended considerably further than the two confirmed sites known today. There is also continuing interest in applying similar analytical techniques, particularly sediment DNA analysis, to museum collections of previously excavated but incompletely studied archaeological material from across Asia, on the theory that additional evidence of Denisovan presence, or additional hybrid individuals like Deni, might already sit unrecognized in existing collections, waiting for the right combination of technique and attention to reveal what they actually contain, much as Deni's own fragment sat unrecognized in a tray of unidentified bone for 4 years before the right screening method finally identified it. This ongoing research effort reflects something important about how scientific understanding of the deep human past actually develops. It rarely arrives through a single, isolated breakthrough that settles every question at once. It develops instead through a slow, cumulative process of careful excavation, technological development, and patient reanalysis of both new and existing evidence, with each individual discovery, however remarkable on its own, functioning as one more piece added to an enormous, still incomplete picture. Deni's discovery was extraordinary, but it was also, in an important sense, simply the most dramatic and clearly visible result, so far, of a much longer, quieter, ongoing scientific effort to understand exactly how connected and how complicated the story of ancient human ancestry actually was. Care across the divide. One of the quieter but genuinely important threads running through this story concerns not just the fact that Deni's parents produced a child, but what her survival to at least 13 years old actually implies about the care she must have received throughout her short life. Care that, if her parents' populations were truly distinct enough to be considered separate species in any meaningful behavioral sense, might not have extended so readily to a child who was, in a very real biological sense, only half like each of them. Human childhood and the childhood of closely related ancient human populations involves an extraordinarily long period of dependency compared to almost any other species on Earth. A human child cannot feed itself reliably, cannot protect itself from predators or harsh weather, cannot acquire the practical skills necessary for survival without many years of sustained care, teaching, and protection from adults around it. This pattern almost certainly applied equally to Neanderthal and Denisovan children, given how closely related these populations were to modern humans, and given the archaeological evidence discussed earlier, showing that Neanderthal communities invested significant care in supporting injured and disabled adults, well beyond any simple calculation of practical usefulness. A population willing to sustain an adult who could no longer hunt or gather effectively was very likely a population equally willing, and very likely far more inclined, to sustain a child, whose eventual contribution to the group lay ahead rather than behind. For Deni to have survived to at least 13 years old, she would have required roughly that many years of sustained feeding, protection from a genuinely dangerous natural environment that included large predators along with brutal seasonal cold, and the gradual transmission of practical survival knowledge from the adults around her. Whatever the precise circumstances of her parents' relationship, and whatever mixture of her mother's community, her father's community, or both, she was raised within, someone, or more likely several people across those 13 years, invested real, sustained effort into keeping her alive. This detail matters because it complicates any simple, cold reading of her existence as merely a genetic curiosity, an interesting biological accident worth studying only for what it reveals about population genetics. She was a child who needed, and by every indication received, enough sustained human care to survive well past infancy and early childhood into the more independent years of early adolescence. This raises a genuinely interesting question that the archaeological evidence cannot definitively answer, but which researchers have thought carefully about. Was Deni raised primarily among a community that would have recognized her as fully belonging, despite her mixed ancestry, or did her mixed heritage create some degree of social complexity within whatever community raised her? Comparative evidence from later, better documented human societies shows enormous variation in how mixed heritage individuals have been received across different cultures and different periods of history, ranging from full, unquestioned acceptance to significant social complication or exclusion. There is no direct archaeological evidence that speaks specifically to how Deni herself was regarded by the community that raised her, and any confident claim in either direction would move well beyond what the physical evidence can support. What can be said, based simply on the fact of her survival to at least early adolescence, is that whatever social complexities may or may not have existed around her mixed heritage, they did not prevent her from receiving the sustained, essential care that any human child, regardless of ancestry, requires simply to stay alive across more than a decade of early life. There is a broader modern echo worth drawing out carefully here, without overstating the connection between a 90,000-year-old life and contemporary experience. Families formed across different backgrounds, different cultures, different communities remain a familiar and often complicated part of human life today, just as they very likely were, in whatever form the specific social structures of the time allowed, during the era when Deni lived. Children born into such families have, across every documented period of human history, navigated their own particular version of belonging to more than one community at once, sometimes smoothly, sometimes with real difficulty, depending on the specific attitudes and structures of the societies around them. Deni's story does not provide any specific answer about how this played out for her personally, but it does offer a quiet, ancient reminder that the human experience of navigating mixed heritage, of belonging in more than one direction at once, is not a modern invention. It is, if her story is any indication, a pattern stretching back at least 90,000 years, into a world so distant from our own that almost nothing else about daily life at the time remains directly recognizable. And yet this particular thread, the challenge and reality of blended belonging, appears to have already existed even then. The cost of what we do not know. Honest science requires sitting with uncertainty rather than resolving it artificially, and Denny's story contains a significant amount of uncertainty that deserves direct, careful acknowledgement rather than being smoothed over in service of a cleaner narrative. We do not know the cause of her death. The single bone fragment recovered provides no direct evidence of trauma, illness, or any other specific cause, and even if such evidence had been present on this particular fragment, a single small piece of one limb bone would be extremely unlikely to preserve clear evidence of whatever ultimately caused her death, unless that cause happened to directly involve significant, visible damage to that exact piece of bone. This means popular retellings of her story that confidently describe a specific cause of death, whether illness, injury, or any other scenario, are moving beyond what the evidence actually supports. Responsible treatment of her story requires acknowledging this gap honestly, rather than filling it with a plausible-sounding invention, simply because audiences generally prefer a story with a clear, specific ending. We do not know whether her body was deliberately placed within the cave as part of any burial practice, or whether the bone fragment ended up in that location through some entirely different process, such as being carried there by a scavenging animal after her death occurred elsewhere, or through natural sediment movement across the many thousands of years since her death. Neanderthal populations elsewhere have left reasonably clear evidence of intentional burial practices at other sites, but the specific context surrounding Denny's fragment within Denisova Cave does not, on its own, provide the kind of detailed archaeological information that would allow researchers to confidently determine which of these scenarios applies in her particular case. We do not know what her relationship was like with either her mother's community or her father's community, whether she spent her life primarily among one group, moved between both, or existed within some other social arrangement entirely that the evidence simply cannot illuminate. We do not know if she had siblings, whether any of them might have shared her mixed ancestry or been born from a different pairing entirely within either of her parents' subsequent relationships. We do not know if she was ever aware, in whatever conceptual framework her community may have had for understanding difference and ancestry, that her parents came from two distinctly different peoples, or whether such distinctions carried any particular social weight within her world at all. Perhaps most significantly, we do not know, and given the current state of the evidence may never know with certainty, the full nature of the circumstances that brought her parents together. Genetic evidence proves definitively that a Neanderthal woman and a Denisovan man produced a child together. It cannot prove, on its own, whether this reflected a relationship built on mutual choice and connection, comparable to how we might understand a relationship today, or whether it reflected different, harsher circumstances more common in the power dynamics that likely existed at various points between different, sometimes competing ancient populations. Responsible researchers studying this discovery have been careful to acknowledge this uncertainty directly, rather than defaulting to either an overly romantic or an overly grim assumption. The genetic evidence is clear. The full human context surrounding that evidence remains, and will very likely always remain, genuinely unknown. This honest accounting of uncertainty is not a weakness in Denny's story. It is, in an important sense, part of what makes her story trustworthy rather than merely entertaining, a version of this story that confidently filled in every gap with invented, emotionally satisfying detail would be easier to tell and might feel more complete to an audience seeking a fully resolved narrative. But it would not be true, and the discipline of holding genuine uncertainty honestly, rather than papering over it with convenient invention, is precisely what allows the parts of her story that are genuinely, rigorously established, her ancestry, her approximate age, the broader pattern of interbreeding her existence confirmed, to carry the full weight and credibility they deserve. Why one bone changed the picture? It is worth closing this deeper examination of Denis' story by returning one final time to the scale of what a single small piece of evidence managed to accomplish, because the disproportion between the size of the physical evidence and the size of its scientific impact is itself one of the most remarkable aspects of this entire discovery. Before 2018, the scientific understanding of interbreeding between Neanderthals, Denisovans, and modern humans rested almost entirely on indirect evidence. The small percentages of archaic ancestry detected within modern human genomes, filtered and diluted through thousands of generations of subsequent mixing and inheritance. This evidence was genuinely compelling and had already firmly established that interbreeding occurred. But it was by its nature indirect. It told researchers about the accumulated statistical echo of ancient unions, without providing a single, direct, unambiguous example of one specific union captured in the act, so to speak, within the genome of an actual first-generation child. Denis' discovery changed that entirely, and did so with a single specimen weighing, quite literally, only a few grams. Her genome did not require complex statistical modeling across thousands of comparison genomes to demonstrate that interbreeding had occurred. It showed the pattern directly, immediately, undeniably, in the clean, unblended 50-50 split between Neanderthal and Denisovan ancestry that no other explanation could reasonably account for. In scientific terms, this represents the difference between strong circumstantial evidence and a direct eyewitness account. Both are valuable, but direct evidence carries a different, more immediate kind of certainty, and Denis' genome provided exactly that, transforming a well-supported theory into an established, directly observed fact. This is part of why her discovery generated such immediate, widespread attention when it was published, reaching well beyond the relatively specialized academic audience that typically follows developments in paleogenetics. The finding was not simply an incremental addition to existing knowledge. It was a genuinely rare example of physical evidence directly confirming, in the clearest possible way, exactly what researchers had long suspected but had never been able to prove with this level of direct certainty. Scientific discoveries of this particular kind, where a single specimen provides such clean, unambiguous confirmation of a previously indirect theory, are genuinely uncommon, which is part of why researchers across the field described her discovery, at the time and in the years since, using language reflecting a level of significance rarely applied to any single archaeological find. Her story also serves as a powerful, concrete illustration of a broader truth about how much information can be extracted from even the smallest, most seemingly unremarkable pieces of physical evidence, given sufficiently advanced scientific technique. A fragment of bone too small and too damaged to identify visually, sitting unrecognized in a drawer for years among thousands of similar fragments, ultimately yielded information detailed enough to identify not only the individual's species and sex and approximate age, but the specific broader population history of both of her parents, down to identifying which other known individuals from the same cave each parent's lineage was more closely related to. This level of detail, extracted from a piece of bone smaller than a house key, represents a genuine triumph of decades of careful, cumulative scientific development, a reminder that the physical remains of the ancient past, however fragmentary and however easily overlooked, continue to hold far more information than earlier generations of researchers ever imagined possible. Somewhere in Denisova Cave, in sediment layers not yet excavated, or in museum drawers holding fragments not yet screened using the latest available techniques, it is entirely possible that more evidence of this kind waits to be found. More direct confirmations of the deep, complicated, interconnected story of how different ancient human populations actually related to one another across hundreds of thousands of years of shared, overlapping history. Denny was the first individual of her kind that science has managed to identify and confirm. Given how much of the physical record from this period remains unexamined, and given how rapidly the technology for extracting information from ancient remains continues to advance, there is every reason to believe she may not, in the end, remain the only one. Chapter 9. What scientists still argue about. No major scientific discovery arrives and settles every question in its wake, and Denny's case is no exception. Even as the core finding that she was a direct first-generation hybrid of a Neanderthal mother and a Denisovan father has been widely accepted and repeatedly confirmed through subsequent analysis, researchers continue to debate a range of related questions that her discovery raised but did not fully resolve. One ongoing area of discussion involves the broader relationship between Neanderthals and Denisovans as populations, and specifically whether it remains meaningful or even accurate to describe them using the language of separate species at all. In biology, the traditional definition of a species often hinges on reproductive isolation, the idea that members of genuinely distinct species either cannot produce offspring together or can only produce offspring that are themselves infertile or face significantly reduced biological viability. Denny's existence, combined with the broader genetic evidence of repeated interbreeding between Neanderthals, Denisovans, and modern humans, complicates this definition considerably. If two populations separated for hundreds of thousands of years can still produce children together, and if at least some portion of those children appear to have gone on to have children of their own, based on the trace ancestry patterns found scattered through both ancient and modern genomes, then the traditional biological boundary that separates species from subspecies, or from regional populations of the same species, becomes considerably blurrier in this specific case. Some researchers have argued that Neanderthals and Denisovans might be better understood not as fully separate species in the strictest biological sense, but as distinct populations, or perhaps subspecies, of a broader human lineage that also includes modern humans, all three sharing enough biological compatibility to interbreed successfully despite substantial physical and genetic differences built up over hundreds of thousands of years of separate evolution. Other researchers maintain that the differences between these populations in physical structure, in genetic distance, and in the broader trajectory of their evolutionary history are significant enough to justify continuing to treat them as distinct species, arguing that biological compatibility for reproduction does not, on its own, erase meaningful evolutionary distinctiveness. This is a genuine, active debate within the field of paleoanthropology, not a question with a single settled answer, and honest science holds that uncertainty rather than resolving it prematurely in either direction. A second area of ongoing discussion involves the frequency and pattern of interbreeding between these populations. Denny's case, combined with the trace Neanderthal ancestry found in her own Denisovan father, provides direct evidence of at least two separate interbreeding events involving these two populations, separated by what researchers estimate to be several hundred generations. But two documented events, however significant, are not enough on their own to fully establish how common such encounters were across the entire span of time and territory where Neanderthal and Denisovan populations coexisted. Researchers continue to debate whether interbreeding was a relatively rare event, occurring only under specific, unusual circumstances, or whether it was a more regular, ongoing feature of contact between these populations whenever their territories happened to overlap, with Denny simply representing the one case where direct physical evidence happened to survive and be found. The broader genetic evidence from modern human populations offers some guidance here, though it does not fully settle the question. The relatively consistent levels of Neanderthal ancestry found across most non-African modern human populations alongside the more variable but still substantial levels of Denisovan ancestry found in certain Asian and Australasian populations, suggest that interbreeding between modern humans and these archaic populations was not a single, isolated event, but occurred on multiple occasions, potentially across different times and different geographic regions, as modern human populations expanded outward from Africa and encountered established Neanderthal and Denisovan groups already living across Eurasia. Whether the same pattern of repeated multi-generational contact applied specifically to interactions between Neanderthals and Denisovans, separate from their respective interactions with modern humans, remains a more open question, one that will likely only be answered as more fossil and sediment DNA evidence continues to be recovered from additional sites across Asia. A third significant area of ongoing research and debate concerns the fertility and long-term reproductive success of hybrid individuals like Deni. As mentioned earlier, Deni herself died too young to answer this question through her own life. But the broader pattern of Neanderthal and Denisovan genetic material persisting within later populations, including within Deni's own father's genome, suggests that at least some hybrid individuals across the broader population were able to reproduce successfully. Researchers studying this question have looked to comparable examples elsewhere in the natural world, where hybrid offspring between closely related but distinct species or subspecies sometimes show reduced fertility, particularly among male hybrids in many mammal species, a pattern that has led some researchers to speculate about whether similar reduced fertility might have affected male hybrid individuals resulting from Neanderthal and Denisovan unions, potentially explaining why so much of the surviving genetic evidence of interbreeding appears to trace more consistently through certain genetic markers than others. This remains an area of active investigation rather than settled conclusion, and applying patterns observed in other species directly to ancient humans carries real uncertainty that responsible researchers are careful to acknowledge. A fourth debate concerns the broader question of what ultimately happened to Neanderthal and Denisovan populations as distinct groups, and how much of a role ongoing interbreeding, as opposed to other factors like competition, environmental pressure, or small population vulnerability, played in their eventual disappearance from the archaeological and fossil record. Some researchers emphasize a model of gradual genetic absorption, in which repeated interbreeding with expanding modern human populations meant that Neanderthal and Denisovan genetic material was not lost, but was instead progressively folded into growing modern human populations over many generations, effectively diluting these populations' distinctiveness until they no longer existed as separate, identifiable groups, even though fragments of their genetic legacy persisted. Other researchers place greater emphasis on competition for resources, environmental and climate pressures, and the particular vulnerabilities that come with small, fragmented population sizes, arguing that these factors likely played the dominant role in bringing an end to these populations as distinct, self-sustaining groups, with interbreeding representing a real but secondary factor layered on top of more fundamental demographic and environmental pressures. Most researchers working in this field today would likely argue that the true picture involves some combination of all these factors, varying in relative importance across different regions and different time periods, rather than any single, simple cause applying uniformly everywhere these populations existed. What all of this debate reflects, ultimately, is not uncertainty about whether Deni herself existed, or whether she was genuinely the daughter of a Neanderthal mother and a Denisovan father. That core finding has been repeatedly confirmed and is not seriously disputed within the scientific community. What remains genuinely open is the wider context surrounding her, the broader pattern of contact, interbreeding, and eventual population change that her discovery illuminated but did not, on its own, fully resolve. Science does not always deliver complete, tidy answers. Sometimes its greatest achievements are not final conclusions, but better, more precisely framed questions. And Denny's discovery gave researchers studying ancient human population dynamics an entirely new set of questions to pursue, questions that continue driving research at Denisova Cave, at Baishia Karst Cave, and at archaeological sites across Asia to this day. Chapter 10. The Girl Who Outlasted Her Own Species. There is a particular kind of quiet weight to standing, even only in imagination, at the mouth of Denisova Cave, looking out over the river valley that Denny would have known, and trying to hold in mind the full scale of what her existence actually represents. She lived and died roughly 90,000 years ago, a span of time so large that it resists easy comparison. If you tried to count backward 1 year for every second, it would take you nearly 3 years of continuous counting to reach the point in time when she was alive. Every human civilization that has ever existed, every pyramid built, every written word recorded, every empire risen and fallen, the entire span of recorded human history, from its very earliest beginnings to this exact moment, fits into a period roughly 20 times shorter than the gap between her death and our own present. She belongs to a stretch of the past so deep that it existed before agriculture, before permanent settlements, before writing, before almost every marker most people use to define the beginning of human history as they understand it. And yet, within that impossibly distant world, she was, in the most literal biological sense, a bridge. Her body carried, in equal measure, the accumulated evolutionary inheritance of two populations that had been separately adapting to life on Earth for hundreds of thousands of years before her birth. Populations that had diverged from a common ancestor long before either fully became what we now call Neanderthal or Denisovan. Populations that had gone on to develop different bodies, different tools, different survival strategies, shaped by different environments across an enormous and largely separate span of geography and time. And then, for reasons the fossil record cannot fully recover, whether through competition over a valuable cave, through the ordinary overlap of seasonal movement across a shared valley, or through some other circumstance lost entirely to time, those two long, separate evolutionary paths converged, briefly, in the lives of two individual people, and produced a daughter. She did not know, of course, what she represented. There is no reason to believe a 13-year-old girl living in a Siberian valley 90,000 years ago had any conceptual framework resembling species, or ancestry percentages, or evolutionary divergence times. She would have known herself simply as herself, a person within a family, within a small community, navigating whatever combination of belonging and difference came with being the child of two parents whose peoples, wherever they came from and whatever they looked like to each other, were clearly not identical. Whether her physical appearance drew comment or curiosity from others around her, whether she was raised primarily among her mother's people, her father's people, or some mixture of both, whether she was seen as fully belonging to either group or occupied some more ambiguous position within her community, these are questions the evidence simply cannot answer, and any confident claim about her lived social experience would be invention rather than history. What can be said, with the full weight of genetic and archaeological evidence behind it, is that her existence proves something profound about the nature of ancient humanity, that no amount of theorizing, modeling, or indirect genetic inference could have established with the same clarity. The boundaries between Neanderthals, Denisovans, and modern humans, boundaries that shaped so much of how these populations have been discussed, categorized, and imagined for well over a century, were never as absolute as the language of separate species implied. These were different peoples, shaped by different pressures, carrying different bodies and likely different cultures across an enormous and largely separate span of time and geography. But when their paths crossed, whether through competition, cooperation, or something more personal, they did not simply observe each other from a safe evolutionary distance. They formed relationships close enough and lasting enough to result in children like Denny, who then lived their own short lives, carrying both inheritances within a single body, before dying and becoming, eventually, the evidence that would prove this entire connected history had actually happened. Her discovery also offers something valuable beyond the specific scientific facts it established, a kind of quiet correction to a much older, deeply ingrained cultural instinct to draw sharp, comfortable lines between ourselves and whatever came before us. For much of the history of paleoanthropology, the tendency was to imagine ancient human relatives as fundamentally other, primitive precursors on a simple upward path toward the achievement represented by modern humanity, distant enough in kind that any meaningful connection or relationship between them and us, or between them and each other, seemed almost unthinkable. Denny's bones dismantle that instinct directly. She is proof that these were not distant, separate branches quietly existing apart from one another until one triumphed and the others faded away. They were close enough in the most fundamental biological sense to become family, and that closeness happened not once in some singular, freakish accident, but repeatedly, across generations, wherever circumstance brought them together. There is something worth carrying forward from her story that extends beyond the specific facts of ancient population genetics, something closer to a quiet form of recognition. Families today are frequently more complicated, more blended, and more surprising in their ancestry than any single generation ever fully realizes at the time. People carry within them the accumulated inheritance of ancestors who crossed distances, crossed cultures, crossed boundaries that once seemed far more solid than they turned out to be, often without any of the people involved fully understanding, in their own lifetimes, the significance of what they were part of. Denny's mother and father almost certainly had no way of knowing that their meeting, whatever its exact nature, would eventually become one of the most significant single discoveries in the entire history of paleoanthropology. They were simply living their lives, navigating whatever circumstances brought them together, the way people across every era of human history have navigated the unpredictable, often unplanned connections that end up shaping everything that comes after them. Denny died young, likely before she had the chance to have children of her own, before whatever combination of Neanderthal and Denisovan inheritance she carried could be passed forward into a next generation. In that specific sense, her own direct genetic line ended with her. But the broader story her existence helped confirm did not end there. It continues today, quietly, invisibly, in the genomes of billions of people scattered across the modern world, carrying small fragments of Neanderthal and Denisovan ancestry inherited from countless other unions like the one that produced her, unions that mostly left no bone fragment behind for anyone to ever find, unions whose only surviving evidence exists as scattered percentages in modern genetic test results, disconnected from any specific ancestor, any specific meeting, any specific story. She is the one case, so far, where a specific meeting between two ancient peoples produced a specific, identifiable individual whose bones survived, whose fragment was excavated, whose collagen was flagged by a screening technique that did not exist when she died, whose genome was sequenced by scientists working in a laboratory built for exactly this kind of impossible task, and whose story could finally, after 90,000 years of silence, be told with real evidence rather than speculation. She outlasted the very population she came from. Neanderthals and Denisovans, as distinct independent peoples, are gone, absorbed and dissolved into the deep genetic background of modern humanity many thousands of years ago. But Deni, or at least the small piece of her that survived, remains, and through her, so does the proof of everything her existence represents. Conclusion. A small piece of bone, no longer than a fingernail, sat in a drawer in Siberia for 4 years, indistinguishable from 2,000 other broken fragments of an ancient world, until a laboratory technique nobody had used for this purpose before flagged it as different. That single flag set in motion a chain of careful, patient scientific work that eventually produced one of the most significant discoveries in the modern study of human origins. Not a skeleton, not a skull, not a dramatic burial chamber filled with artifacts, just a splinter of bone, and inside it, a genome that proved something scientists had long suspected but had never been able to directly demonstrate. Her mother was Neanderthal, descended from a lineage that had crossed vast distances across Eurasia, carrying within her the accumulated adaptations of a population that had survived Ice Age winters, cared for its injured and its elderly, buried its dead with what looks very much like intention, and thrived across an enormous range of environments for hundreds of thousands of years. Her father was Denisovan, a member of a population so poorly understood, even now, that researchers can say more about the survival advantage his people left behind in modern Tibetan lungs than they can say about what his own face actually looked like. Two people, from two different, deeply diverged branches of the human story, met somewhere in or near a cave carved into the Altai Mountains and had a daughter. That daughter lived a short life, likely no more than 13 or 14 years, in a world defined by cold winters, seasonal movement, small close-knit communities, and the constant quiet work of survival that shaped every day for every ancient human population across this era. She died, and her body, or at least one small fragment of it, ended up buried in the accumulating sediment of a cave that had already been sheltering different populations of ancient humans for hundreds of thousands of years before she was born, and would go on sheltering others for tens of thousands of years after her death. For 90,000 years she waited, not consciously, not with any awareness, simply as inert matter, slowly transforming under the pressure of accumulated time, mineral gradually replacing organic tissue, the fragment surviving where so much softer material around it did not. She waited through the entire span of recorded human history, through every civilization that would ever rise, through the invention of writing, of agriculture, of cities, of everything modern people generally consider when they think about the story of humanity. She waited until 2012, when archaeologists working a careful, methodical excavation in Denisova Cave's East Gallery pulled her fragment from the ground along with countless others, unaware of what they had actually found. She waited 4 more years in a drawer, until a molecular screening technique identified her as human rather than animal. And then, finally, in a laboratory in Germany, her genome was read, and the silence of 90,000 years broke all at once, into a discovery precise enough and clear enough to make headlines around the world within a single day. What her discovery ultimately proved was not simply that interbreeding between ancient human populations occurred, something researchers had already established through less direct genetic evidence years earlier. It proved something more immediate, more personal, and in its own quiet way, more moving. It proved that 2 different kinds of ancient human beings, separated by hundreds of thousands of years of independent evolution, by different bodies, different adaptations, different histories written into their very bones, could still come together closely enough and stay together long enough to become a family. Not a distant genetic echo scattered thinly through thousands of generations of later descendants, but a direct, immediate, undeniable union, preserved exactly as it happened, in the body of the child who resulted from it. The evidence remains honest about its limits. We do not know her name. We do not know her face. We do not know whether her parents' relationship reflected choice, circumstance, or something closer to the harsher realities that likely shaped contact between different, sometimes competing populations in the ancient world, and responsible science holds that uncertainty rather than resolving it with comfortable invention. We do not know how she died, whether suddenly or slowly, whether surrounded by people who cared for her or largely alone. What we know is narrower, more precise, and in its own way, more powerful than any invented story could be. We know her ancestry, confirmed through repeated, rigorous genetic analysis. We know her approximate age at death. We know, with as much certainty as ancient genetics allows, that she was real, that she lived, that her existence was not theoretical but actual. Her discovery reshaped how researchers understand the broader relationship between Neanderthals, Denisovans, and the modern humans who eventually inherited fragments of both populations' genetic legacy. It confirmed that the boundaries separating these ancient peoples were never as rigid as the language of separate species suggested. It revealed, through her own father's genome, that this was not an isolated event, but part of a longer, quieter pattern of contact between these two populations, stretching back further than her own birth, continuing to echo forward through the genomes of billions of people alive today who carry small percentages of Neanderthal or Denisovan ancestry without ever knowing the specific unions, the specific meetings, the specific individuals responsible for that inheritance. Denisova Cave still stands in its river valley in the Altai Mountains, still holding, somewhere in its remaining unexcavated sediment, whatever additional fragments of this ancient story have not yet been found. Baishia Karst Cave still sits on the edge of the Tibetan plateau, prayer flags marking its entrance, holding its own share of Denisovan history within layers of dirt that researchers continue to study using techniques that grow more sophisticated with each passing year. Somewhere within those places, and within other sites not yet excavated, or not yet even discovered, more pieces of this story likely remain, waiting the way Denny's own fragment waited, for the right combination of careful excavation, patient laboratory work, and scientific curiosity, persistent enough to keep looking closely at what might, at first glance, appear to be nothing more than an ordinary, unremarkable piece of broken bone. She was 13 years old, the daughter of a Neanderthal mother and a Denisovan father, and for 90,000 years she was the quietest kind of proof imaginable, sitting patiently in the dark, waiting for someone to finally understand what she had been carrying all along.